Hello student, today we start the BSc third year, that is paper number 13, aquaculture technique and fish nutrition. Aquaculture technique and fish nutrition. Within that, unit number 1 already we are completed, that is the fish culture. Unit number 2, that is marine water prawn culture. We are going to that topic, that is the marine water prawn culture. Within that, study of general characters, food and feeding habits, selection of sites, collection of brooders. Already we are completed. The 4th point in the marine water prawn culture already we are completed. Then the next 5th one, that is mating, spawning and development of marine prawn. Mating and spawning means the prawn, when we culture the prawn, that time the important thing is that is the spawning season, that is the developmental stages of the prawn more are more important. How can they mate, how can they spawn? We will see this topic, the title, that is the mating and spawning of marine prawn. After the collected stock has reached the destination, first point, then the prawns are released into the tanks containing fresh seawater for the purpose of breeding, for the purpose of mating. It's very initial, that is the fresh water, seawater is very important, fresh. Okay. Thereafter, they have recovered and become active. After that, fresh seawater. After that, the procedure, they have recovered and become active. They are transferred to the bigger pools, about 250-liter capacity, for spawning. All the process is going for the mating and the spawning. After that, we will see about the development. First of all, we see the mating and the spawning point. They are transferred to the bigger pool, about 250-liter capacity for spawning. Okay. Then plastic bag, and the procedure for the purpose of spawning. Plastic line poles printed black on inner side of those of the cylindro-conical fiberglass are usually recommended. This is the type of cylindro-conical conical fiberglass are usually recommended for the purpose of spawning mating. Okay, only one pair breeding purpose, that is one male and the one female. Only one pair are kept together in the tank, and a volcano screen is mounted at the open end and check escape jumping out the tank. Mating take place in the tank. This is the process of mating and the spawning take place over the prawn. When we culture, the process is adopted. Okay. Then next one, mating, spawning, the last bit, that is the development. How can they develop after the fertilization, after the breeding, after the spawning? The next point, that is the development. The time taken for copulation varies with the species. Okay, it takes 3 to 4 minutes in Peneus japonicus, one type of marine water prawn, and 80 minutes in Peneus monodon. Already we know about the Peneus monodon, Peneus indicus. They are the famous for the Indian prawn culture. Two example, that is Peneus japonicus and Peneus monodon. Okay, spawning take place only after the eggs are mature. The important point that when the spawning take place after the egg is mature. In monodon, about 7 lakhs are shed in about 3 to 4 minutes. Okay. Then simultaneously spermatophores are discharged so that fertilization may take place externally in the water. Fertilization may take place externally in the water. Fertilized eggs soon slide to the bottom and larva hatches after about 8 to 9 hours or 10 to 12 hours. The larva is called the nepulis. Important point. After 8 to 9 hours, the formation of nepulis. It metamorphoses and changes into the adult prawn. Then the period of metamorphosis is the most crucial, during which various physicochemical factors should be watched and regularly cared for. The important point is that after the fertilization, after the nepuli, when we culture the prawn, the process is going on. The important thing is lastly, that is the physico-chemical factor. What is meant by the physico-chemical factor? That is about the water quality, about the temperature, about the intensity, about the salinity. All are important for the purpose of prawn culture. Okay. Thank you.